Welcome to our tutorial on performing circuit queries on transcriptomics data in braintrawler. To begin, we first create a circuit on the Circuit Creation page. We choose the first Circuit Creation option, which allows us to load a predefined circuit from the braintrawler database. After selecting the circuit, the nodes are listed on the left and visualized on the right. This particular circuit represents brain regions associated with the RDoC, or Research Domain Criteria, domain negative valence. Let's now navigate to the Circuit Query page. Our circuit is already selected, and we now need to select a transcriptomics dataset. Let's go for the Allen Human Brain Atlas. Next, we can choose from 6 different query types. Each query type is essentially a t-test comparing the expression between different spatial selections. For example, you can query for differences between specific circuit nodes and the rest of the brain. For now, let's compare the expression in the circuit to the rest of the brain by selecting Circuit versus Brain. After optionally limiting our search to specific cell types or subject categories, we can execute a one-tailed or two-tailed test by clicking on Higher Expression or Differential Expression. The query result below shows a list of genes, ranked by their Q-value and fold change. These values are also shown in a volcano plot above the table. Here, you can set filters for Q-value and fold change to narrow down your result list. Filters for sample metadata, such as cell type, need to be specified before executing the query. Each row in the result list corresponds to one gene, showing gene metadata, such as ensemble ID and gene short name, and detailed query results, such as mean expression in the compared regions, Q-value and fold change. The entire table can be exported to a .csv or .tsv file by clicking the download button above the table. By clicking the eye icon on the left of a gene entry, its expression is visualized in the renderer. You can toggle the visibility of items in the renderer from the workspace to prevent occlusion. You can find additional information on the braintrawler help page. Thanks for watching.